shaping policy for ethics and genetics from the 1990s and beyond. And it's an example of an international ethics committee. So in our course on IRBs, we've had the lecture today on the IRB regulations and abuses and compliance issues in the United States. In the last 2 or 3 weeks, we've looked at UNESCO reports on policies and procedures for different types of ethics committees. Remember, there were policy-making committees for the government level. There were professional association ethics committees. There were hospital ethics committees, and there were research ethics committees, the general types. This example, the Human Genome Organization Ethics Committee, I'm going to talk about one aspect of a professional association ethics committee, and I'll also refer to international ethics committees in this discussion. So there are international standards for bioethics and genetics. The Universal Declaration on the Human Genome and Human Rights was the first international standard for bioethics adopted by both the UN General Assembly and also UNESCO. It means all the member countries agreed to comply by the recommendations. And in 2003, the International Declaration on Human Genetic Data was another such document. So all member countries adopted these declarations. It means they commit themselves to comply. It means all institutions in the countries should comply by the principles. I'm going to talk about the Human Genome Organization as an example of a professional association statement. But there are, of course, other general ethical guidance documents for regulating genetics practice. Genetics was a catalyst for ethical reflection. The responsibilities as scientists, both social and natural science in general, were revisited. And we saw this, the first declaration on bioethics was because of the Human Genome Project. It was seen as an issue in the 1990s which could bring all the countries of the world, despite their different cultures and practices, together to agree upon a document. The statement includes various articles. For example, Article D13, the responsibilities inherent in the activities of researchers, including meticulousness, caution, intellectual honesty, and integrity in carrying out their research, as well as in the presentation of utilization of their findings, should be the subject of particular attention in the framework of research on the human genome because of its ethical and social implications. Public and private science policymakers also have particular responsibilities in this respect. Now, the Human Genome Project, the first human genome to be sequenced was that of a man called Craig Venter. That was the first complete human genome sequenced. The cost of that sequence was around $3 billion US. The cost using the latest DNA sequencing of the cost of sequencing any one of your DNA completely, the genome of Ramesh, it would cost $999. But a company would charge you $10,000 US. But that's the actual cost. The actual cost of reagents and the sequencing, they use a different technology now, is under $1,000 a genome. So thousands, in fact tens of thousands of people have had their complete human genome sequenced now. You can get the information. To analyze it actually will cost much more than the actual sequencing. Now, meticulousness was important for science, but now what's science? Well, science is increasingly motivated by the philosophy of wisdom rather than the philosophy of knowledge. Namely, we pursue science in order to achieve some benefits for our community. Researchers are expected to involve the community in decision-making about the planning, conduct, and feedback of a proposed research. And community engagement is the mutual communication to discuss about the project with a wide range of people in the society to better understand societal concerns and provide an opportunity for broad public input. The practical mechanism of community engagement will differ with the specific genetic research being pursued. The general concept of community engagement must be applied to different situations and countries. The efforts are more than one-way education from researchers, but involve a process of interactive bioethics between the different parties in the community. And the principles of community engagement include consultations with tribal communities, which we discussed, or with another interested party. The HUGO Ethics Committee, the Human Genome Organization, was a group of scientists who conducted the Human Genome Project. So the leading scientists in genetics and genomics became members of this organization. An ethics committee was formed, and they released a series of statements, and some of their statements are here, and they were relatively important in terms of setting the international guidance for human genetics. The principal conduct of genetic research, statement on DNA sampling, who has control and access to this, statement on cloning, especially looked at issues such as mitochondrial cloning, which were actually possible at that time, benefit sharing, and the human gene therapy. In the principles of the HUGO statement on principal conduct of genetic research, we see recognition of the human genome as part of the common heritage of humanity. And this actually was an initial statement in the drafts, the 9 drafts of the Universal Declaration of Human Genome and Human Rights. It was dropped, weakened substantially in the final adopted text, saying in essence, it's like the heritage of humanity. The reason for the droppage was, if the human genome is part of the common heritage of humanity, you cannot patent it. Okay. That at the time in the 1990s was intense debate on the patenting of the human genome data. Adherence to international norms of human rights, respect for values, traditions, culture, and integrity of participants, and acceptance and upholding of human dignity and freedom. In the 1990s, mid-1990s, saw a number of issues, both of the Genome Project, the Human Genome Diversity Project, attempts to map the genetic differences in different tribes and populations, and many other issues. The members of the committee were philosophers, lawyers, and scientists from actually 12 different countries. And many of them have been very active in international ethics and bioethics. And so the chair of the committee in its very active years was Professor Bertha Maria Noppes, Canada. She's in the University of Montreal. She was a Canadian Scientist of the Year, even though she's a lawyer. And she's still very active in international law. Ruth Chadwick then became the chair. She was very active in the UK. And people from different aspects of Justice Michael Kirby. These are also, if you look at the papers written by these people, they're all influential. All of them have written hundreds of papers. Each one of them has written hundreds of papers on bioethics and genetics. Justice Michael Kirby was a High Court Justice of Australia. When he retired, he then became the sort of ethics and science advisor to Her Majesty Queen Elizabeth. He led to, in fact, reviews of many substantial documents around the world. And he was Human Rights Commissioner for a number of doubtful countries in terms of investigating their human rights abuses. So some excerpts from the types of statement or article that regulate types of science. It's just to show you the sort of language that people adopt as a regulatory statement for science. So on the DNA sampling, the choices offered in the consent process should reflect the potential uses of the DNA sample and its information. It's important to indicate whether the sample and its information will identify the person, code the identity, or anonymize the identity so that the person cannot be traced, although some demographic and clinical data may be provided. Even if anonymization is appropriate in certain circumstances in research, caution should be exercised in any irreversible stripping of identifiers from the samples, since it may preclude valuable use of the samples and validation of results. Now, this is for samples that, in the words of what Tom showed, fluids and tissues that are removed. But the same principles also refer to behavioral data and how do you code or deal with privacy. So for example, if I'm doing the research of the DNA samples of everyone in this room right now, I may take initially the informed consent from all of you. And assuming everyone agrees, then you sign a informed consent form with your name. We give that informed consent form number, and we might then take the blood. Now the blood may be put into a tube, and the tube may be then given an alphabetical code. Somebody might, if we want to keep that sample linked, then make a coding sheet that lists the number of the consent form with the random code that's given to the tube, and that sheet will go to the chair of the IRB and be kept in a locked safe for as long as the law requires in that country. But that coding sheet then will be locked, and maybe in a sealed envelope as well as in a locked safe. Now, that's a coding sheet, and that would be a way of keeping a coding sheet. So then the tube has a random identifier which would then be used in all the scientific research. If I decide to anonymize the identity, it means I'm going to not keep a piece of paper with a list of numbers of consent form and the code on the tube. There's not going to be any such paper. It means from that day on, there's no way, except for complete genome sequencing, to tell me what sample relates to one person. And that becomes totally anonymous. Now it means if I suddenly find in my analysis of that sample somebody had imminent threat to their health based on their genetic sequence, I can't
let them know.
I can't let them know. I could tell, well, this group of 18 people, which I do remember, one of you, by the way,
Scare the hell out of everybody.
is going to die next year. And if you're interested,
You shouldn't go on a long trip.
Or this is your last chance to go on one.
and if you're very interested, you better finish your master's thesis right away. But you could then do the genetic test for that specific disease, which might only cost you $50 or $100, and then you can actually find what we'd found. To get the complete genome sequence, as I said, would cost you $10,000 for a commercial lab, or $1,000 if you were friends of the owner of the human genome.
And the percentage of
DNA
those who would have a
analysis.
heart attack in the process of you notifying them of such information. What about those? That's a whole other study in and of itself.
That's another study. So this is the identifier. Now the advantage then of keeping the identifier, let's say it's for medical research, is that if I keep the identifier in a few years' time, we actually have a very precise and very useful predictive test if we had in the consent form the words, in 4 years' time, if this research does find something that is going to be able to predict, you could list a percentage or certainty that I will suffer, do I want to be notified of that result? And they said yes. We would then take that data with the code, and the researcher would send it back to the IRB, and the IRB chair may open the sealed envelope and then check if they had actually given the consent, and if they had given the consent, there could be a follow-up. And so at the time, the NIH of the United States at this time, and in the 1990s, early 1990s, was saying there should be no way to follow up with people. But the Human Genome Organization Ethics Committee, we thought, in the spirit that people give the research sample to the study, a reasonable person may want to know if they're going to get sick, and it would be a reasonable scientist would tell the results back. So in fact, keeping a coding sheet or some method of including this would be reasonable. So here we had, some national committees in their regulations go through changes in the extent to which they'll inform people. And so that was a sort of a interesting, we were in a sense slightly radical, and we felt that we could be, being all quite well-known ethicists and on different national ethics committees, that we saw some problems in certain national ethics committees. So that was why you had these sort of statements to say, well, think again to NIH and think again to certain national committees about the way that your ethical duties to participants. Now, security mechanisms should be put in place to ensure the respect of choices made. And international standardization of ethical requirements for control and access of DNA samples and information is essential. Here again we saw there was so much variation. Another example of the statement on DNA sampling, control and access is routine samples obtained during medical care and stored may be used for research if there is general notification of such a policy. The patient has not objected, and the sample to be used by the researcher has been coded or anonymized. Routine samples obtained during medical care and stored before such notification of such policy may be used for research if the sample had been anonymized prior to use. So here was an issue. Samples, for example, a liver biopsy from a patient taken in 1968, if that was anonymized, could be taken. Now there are interesting issues. That was a routine sample from medical procedure. Research samples is a sample taken for research, not for therapeutic medical care. Obtained with consent and stored may be used for other research if there is general notification of such a policy. A participant has not yet objected. The sample to be used by the research has been coded or anonymized. For the use of research samples obtained before notification of a policy, these samples may be used for other research if the sample has been coded or anonymized prior to use. Okay, so in this case, coding was accepted as well. So that means if a coding sheet was kept in the safe of the IRB, it could be used. Of course, if the sample is anonymized and there's no coding sheet, you can't actually tell anyone, but you can use the samples for taking let's say, doing research. So let's say in the famous or infamous research studies that were done on the orphan tribe on diabetes in the 1960s, depending on the type of consent given for the taking of their samples, somebody might go to Phoenix Medical Center freezers and then look at the samples and consider whether they could use those samples or not. And you could think of some research for the general population that might be useful. For example, let's say we find perhaps there's some damage caused by mining in the last 30 years to the DNA of the general population living downstream. having the samples in 1968 would allow us to then look at the DNA mutations that occurred because of the impacts of mining. And in fact, the original aim of the original aim of the Human Genome Project, which you will find very interesting in 1989, the original aim of the Department of Energy of the United States, of why it joined the National Institutes of Health, is that the only way we can test a mutation at low level from environmental radiation is by total genome sequencing. And that's actually what they've accomplished. Now, it's now 2014, it's 25 years. Now the cost of sequencing a human genome is under $10,000. It becomes actually reasonable. But see, in 1990, when they started, the cost estimate was around $3 billion per genome. And it was $3 billion dollars. But now it's $10,000. Actual real cost is now about $1,000. So now it becomes possible that 25 years later, thanks in a sense to the foresight of the Department of Energy people for doing their radiation and mutation, environmental mutation study, they can now do it in a research study. Now, if we have those samples, who will give consent for the use of those samples? The individuals, depending on the informed consent, may have given. Let's say it's of the general population of Phoenix, probably no consent is needed. If it's of a particular tribe, the consent of the community representatives of the tribe now are also needed. Leanne, you had a question.
Well, first, initially you said they gave their blood for diabetes. And then now you're testing it for mining?
For mutation of
For
mining.
mutations in mining. Wouldn't you have to go back and get another consent?
It depends on the way the consent was framed.
And I bring this up because they have the ASU lawsuit with the Havasupai tribe that
Yes.
they were testing for schizophrenia.
Yeah. This was, the Havasu tribe consent forms were done much later, when consent forms became much more specific. And so the ASU researchers had a very narrow consent form. And they broke the terms of their consent form by doing schizophrenia linkage studies on studies which should have been done on only a particular disease, diabetes, they say.
Wow.
Okay, so that's why the ASU fired their researchers and they paid compensation. But in the 1960s, the consent forms were often very general. Now, this actually could turn out to the advantage of the community, but what we need is to see the actual consent forms, what they consented to. But if they consented to a very broad medical research, then their samples could be used as baseline data for comparing the impact of chemical mutanants in the environment from 1960s to 2010 or 2015, or when we do the study. Okay. So this is, depending on the consent form, quite relevant. Okay. So, what does it say about access by relatives? Well, special consideration should be made for access by immediate relatives where there is a high risk of having or have transmitted a serious disorder or prevention or treatment is available. Immediate relatives should have access to stored DNA for the purpose of learning their own status. These exceptional circumstances should be made generally known at both the institutional level and the research relationship. Now, so this again was saying the reasonable family, if somebody gives a sample and they find they have medical risk, and then that person dies, most family members would want to help their children or their brother or sister, and it should therefore be reasonable that this access may be granted. And again, this was countering a language at the time which was saying only that person could give and family members could not get access. And again, it's saying, well, what's reasonable? Reasonable families, if your children is going to benefit from the use of your sample that collected from you in the past, reasonable parent will want the children to benefit. In the absence of need of access by immediate relatives, stored samples may be destroyed at the specific request of the person. Such destruction is not possible for samples already provided to other researchers or entered into a research protocol used for diagnostic purposes. By their very nature, anonymized samples cannot be withdrawn or destroyed. If you anonymize it, you protect the person. But you actually also, one thing you can never do is you cannot never take your sample back. It's anonymized. Nobody knows which is your sample. Unless authorized by law, there should be no disclosure to institutional third parties of participation in research, nor of research results identifying individuals or families. Like other medical information, there should be no disclosure of genetic information without appropriate consent. Okay, so these were examples. What about benefit sharing? Benefit sharing was interesting. A statement appeared in the journal Science, which is a major scientific journal, because it's very topical at the time, especially relating to tribal populations giving samples. So our recommendations were that all humanity share and have access to benefits of genetic research. That benefit not be limited to those individuals who participated in such research. That there be prior discussion with groups or communities on the issue of benefit sharing. That even in the absence of profits, immediate health benefits as determined by community needs could be provided. That as a minimum, all research participants should receive information about general research outcomes and an indication of appreciation. And C, that profit-making entities dedicate a percentage, 1 to 3% of their annual net profit to healthcare infrastructure or humanitarian efforts. And it's very interesting. This, for Article 6, was, we decided, we actually visited the major pharmaceutical companies doing genetic mining. We went to the labs of, in fact, 4 or 5 of the major pharmaceutical companies in the world. And in general, they agreed with us. And so, in fact, in my experience with discussing with Big Pharma on ethics, their dilemma was how to deal with corporate social responsibility and how to provide some reasonable return. And so many of them decided they will set up initiatives like a tropical disease research institute. And that was a way, if they give, a few $100 million to general research focused on tropical diseases, this was a way to provide humanitarian access and would probably, in our opinion, this committee, and in fact the same as I knew, provide a better return for general humanitarian efforts than some written statement that we'll give you 20% of the profits from this drug if the drug is ever profitable, because most research is never profitable. Okay. So that was our tendency, and the reason we also thought the benefits should be shared is that many genetic research, the sufferers of genetic disease are across every population in the world. That you might do a study, for example, on diabetes in a particular tribe here. The diabetes sufferers having that genetic mutation are all around the world. So there's some sense of benefit sharing to every sufferer who has that disease. And of course, individual communities could negotiate, we're going to get a health center for our work. But in general, those sorts of health centers or research enterprises were going to be much better for the tribe than any percentage of return on some future drug, because most, as I said, almost all research doesn't lead to a profitable drug. So how do we implement this? Well, there's very many challenges. One is low awareness. People are not aware. We need often a new technology to catalyze reflection. By the way, this is a display I made I had a museum. I have several museums of bioethics. I had one in Bangkok. There's still one in the Philippines, and there's several others. And I actually got the idea from the French National Science Museum of cloning, that you have lots of babies in them. And I had one of my research volunteers go and, he was a guy, a Japanese guy. He went to the market in Bangkok and bought 20 baby dolls, and the shop thought he was rather kinky to buy baby girl dolls, 20 of them. But we thought this is the best way to make this display. And all sorts of issues, catalyze our reflection. We have energy ethics, clothing ethics, space ethics, animal rights, nanobiotechnology, genetic medicine, population genetics, all sorts of things. But genetics was clearly a big catalyst to reflection on ethics, because many of the principles apply to all research. Implementation of genetics bioethics standards. In my past life in UNESCO, one of my jobs was to implement these declarations. We made national translations with many national governments because states committed to take appropriate steps to provide a framework for free exercise of research on the human genome with due regard for principles set out in the declaration. Now, this is two sides of the research equation. One is guaranteeing free conduct of research. So scientists are free, genetic researchers, social scientists, but also thinking of the legal principles, human rights, fundamental freedoms, human dignity. When we thought about samples, most biological samples are taken as clinical practice and diagnostics. So when you take a sample from people, if you go to take a test now, you often need to take the sample. One of the declarations principles. Go to pass me on water.
Should I get you a water?
Thank you. No, I've got one. Thank you. So, now genetic data should be held confidential. What other types of data would be held confidential? What's held confidential? Think of some other data, if I collected from you, that should be held confidential.
Your name,
Social security numbers,
anything to identify the person.
Okay, so let's say AUSN is it's keeping a record of its students. Do you think the name of the students will be held confidential from the federal government?
No.
Well, you're talking about for research studies, though.
Yeah.
Yeah. They want to do research. Or it says research or any other purpose.
Oh.
Any other purpose. But it includes anything else. So some things like a name may be not kept. We may see the report of names. But your HIV status, would this be kept confidential?
Yes.
What say you want to apply for immigration to some countries? The country may require it, but the organization will not disclose it. Your criminal record? Do I keep the criminal record of all our students confidential? No.
Do we have a criminal record of all our students?
We do not ask the question. So the only place on our application form when people write, I decided to study bioethics because after my last 5 convictions for murder, I seen the light. So that sort of information, do I need to keep it confidential? Well, probably as an ethical school, we would keep it confidential. However, if it was an investigation into the murder and the sudden appearance of our students from our classes, the law in the country might require our disclosure. And also a reasonable professor might start to look back at his application form. So if you give me the data that you are criminal, I may keep it confidential, but we will use a,
There was, remember in Virginia, the mass killing of the
Yes.
students, so the teacher, people were blaming, I don't know what had happened, but I saw in the news that the student did write in the essay that, how, something about killing or something, but the teacher did not, she thought it's only an essay, so she never guessed that the student is going to, after a few months, kill so many people, and people were debating that she should have informed, and how would a teacher inform, so we all every day give so
Did
many
they-
essays to, yeah.
Did they hold that teacher liable in any way?
I saw afterwards, I don't know, I didn't follow up, but in the Virginia University case, yeah.
So we have a number of questions about
How-
privacy, yeah, and in fact we've had a whole lectures on privacy. It's a big topic. Is it national or global? What about the rights of individual privacy, familial privacy, community privacy, ethnic group privacy, access to information by others? So how do we find the right balance between beneficence of the donor's intention to contribute to research and the privacy protection extended by law? Because you have conflicts. Some individuals will say, I want to give my sample for all research. And others will say, in the community may say, no, well, that's the private information we shouldn't share. You also, for example, you do social studies research, and you find the traditional knowledge practices of a community, and this person says, yes, you can publish it. Another person says, no, you can't publish it because it's our sacred knowledge. If we're going to put our thesis, master's thesis, we may in the end have a public version if we put our master's thesis online as a public document, and we might have some parts which are private. This is something which we haven't had to deal with, but we may have to. And I've seen cases where we do discussions with where some parts to protect the privacy, we want the same for the research publications. The limitations to principles of consent and confidentiality may only be prescribed by law or compelling reasons within the bounds of public international law or international law of human rights. So the limitations to this should be outlined in law. In some countries there is not law, national law, so you use public international law or international customary law through all the declarations. So all the declarations you saw in the lecture on IRBs by Professor Jonas are part of customary international law that applies even if the country doesn't have a specific law. There's an international custom. I want to give now an example of a project I was involved in on community engagement. Now, of course, the issue of community engagement will differ with the specific genetic research being pursued. The general concept of community engagement must be applied to different situations and countries. And the efforts are more than one-way education from researchers, but involve interactive bioethics. This, if you're interested, is a cover of some publications related to the Human Genome Project on the journal Nature. Journal Nature is the #1 most famous journal of science. It's been going for almost 400 years now, and it's published a lot of cover pages on the human genome. I'm going to talk in particular about this one, the Hapmap Project. This is a paper by the International Hapmap Consortium. There's a number of these papers. I was one of several hundred members of this consortium, and it was one of the agreements of the consortium that everyone will be an author on the paper. So it was a good way to get a lot of publications, but in good science journals, in fact. So we did a paper on the community engagement and informed consent, because this process was quite interesting. Now, what was the Hapmap Project? Hapmap was an international collaborative project collecting blood samples from people in various parts of the world. In this project, initial project, there were 240 DNA samples from 4 populations initially analyzed to develop a human haplotype map. It did not collect names or any kind of health information, but researchers will know only what group each sample came from. There will be no direct benefit to the donors except the future possibility of medical research results in general. The project raises ethical issues in the way that the samples are collected, the way that samples are sent to the central repository, and then stored as cell lines for access by researchers around the world, as well as results are put in a public database. So one of the samples was from Japan, and I was a member of, in fact, responsible for collecting and getting informed consent from the Japanese population for this. And Hapmap samples, the cell lines and DNA, are stored in a repository called Coriell repository. It's a private repository, but anyone in the world can apply for the use of the samples. And the haplotype map samples are the basic population samples for, Caucasian sample, Euroban, which represents the African sample, the Japanese sample, and the Chinese sample. Okay. And so this were the initial. Now later there's been further application of Hapmap samples, but this was an interesting case, and I'll give this case. Bioethics Institute is my NGO, and one of my employees, Eiko Suda, worked for me on this. We were paid by this project to do the sample collection. And in fact, Eiko got a PhD based on the samples from here. So we had an ethical legal working group. We did public consultation, public forums, focus group meetings, and recruited volunteers. Somewhat related was a community advisory group, which still exists. I'm still a member of this group in Japan. And it's there forever to sort of, if there are difficult ethical challenges that may cause problems with the Japanese population and the genetic research that's being done. The University of Tokyo was responsible for blood drawing, means taking the blood, and informed consent, though they delegated us to get the informed consent. Shinshu University was to establish the cell lines from the cells, so these cells are immortalized. You get the cell from people's blood, you take out the DNA, you also make a cell line. The cell line will be immortalized so people can use that cell as a cell line. So in fact, there's a set of Japanese cell lines which people can get. Coriell cell repository, and then the analysis came back from the USA back for Riken was part of a global program, analyzing particular sequences, particular chromosomes on all samples from different plants. So we had an interesting, some events, for example, in the IRB protocols. We met on assignment of task in April 2002. We prepared documents, consultation, discussion in Japan. We had teleconferences and meetings. We had meetings with the Hapmap groups going on in NIH. And in the US, a lady called Dr. Ellen Clayton at Vanderbilt was working on the program, as along with the head of the NIH ethics section, who's still actually there, Jean McEwen, still in NIH. They came, we tried to kind of negotiate with the Japanese research ethics community what would be acceptable for between the NIH standards. So it talks about how you would get the protocol approved with both the NIH, with Japan Ethics Committee, European Ethics Committee, European community in a center in Nigeria, and one in China. How would you get 5 committees approving the same informed consent document? Well, we did, but some give and take was required. Then we had the Japan Society of Human Genetics, and then we had these draft protocols. They were actually approved towards the end of that year. So the process of getting the informed consent documents took about 6 months to get each country agreed in the different back translations of documents. And eventually in April 2003, 1 year after the initial meeting, we had the Tokyo University approve. And then we could collect the samples before the money ran out in August, which was actually an issue, that the IRB process was taking a year, and the Japanese government gives the research for this year, but you have to use it by this month. You cannot, they do not carry it over. What was interesting was that initially Japan insisted, partly for national pride, because they're giving 25% of the global funding. We have the technology to make our own cell lines from the blood samples. They did. Unfortunately, when they sent them to Korea, the whole Japanese set was contaminated by mycoplasmas. And we had to actually collect another set. Because we had we didn't keep the blood samples. So we actually had to collect twice as many samples as we initially thought. We had to do the process twice. Sorry for the sound. Community public consultations included a lot of meetings, public meetings, academic meetings, meetings also internationally at Human Genome Association, Asian Bioethics Association. We took videos of public meetings to record the community engagement. I still have some of these videos. Some of the issues raised. Labeling of samples. Should the sample in Korea be labeled as Japanese or East Asian? And in the end, it's labeled as a sample of Japanese people living near Tokyo. It's the formal label, because that describes what it was. Because it was not only, it was people living near Tokyo. We didn't do sample collection in Osaka. We did sample collection only in 2 or 3 locations, and these were all around Tokyo. Abuse of public data. People were concerned that public data would be abused. That maybe minority groups in Japan might be discriminated. Now that there's a global sample representing Japanese group, what happens when they find that this group has increased frequency of a certain genetic disease? This will mean anyone with Japanese ethnic origin can be discriminated because Japanese people have a particular genetic disease. And discriminate Japanese minorities overseas. We're particularly thinking about the Americas, about, for example, Brazil or United States, where there's significant Japanese minority populations who could be discriminated if, at this time, insurance companies might discriminate based on the risk. The meaning of community engagement, we changed the term. How do we confirm the participants were actually real Japanese and not hidden Koreans? Now the issue of hidden Koreans is interesting after the occupation of Japan and when the war ended in Korea. Many Koreans came to Japan. Now in order to avoid discrimination, Japan only allows you to have one passport, and so does Korea. So you have to choose one or the other. And some Koreans chose to take Japanese names and Japanese passports, and some keep their Korean passport. We also have these different, biologically, Japanese people mixed with German people from the Asian continent in Mongolia about 3,000 or 5,000 years ago. Is a sample good enough for the mapping? We actually, in the end, have I think around 70 samples in the repository. International ethical standards versus Japanese norms and laws. Continue to discuss this in many meetings. We also did research. I looked at research I'd done on the concepts of individuality, the race, Ainu culture, for example, in Japan. Ainu, the real Japanese, or the introduced Japanese, the real Japanese. Colonization of Hokkaido. How do we understand the Japanese community? And Most of the community engagement research was actually not funded from Hapmap. I used other funds because I still wanted to do it. We had to modify original plans according to community concerns and interests. So, for example, labeling. We didn't want to label in the end East Asian sample. The Japanese and Chinese samples were labeled separately. Japanese living near Tokyo and the Han Chinese, were actually taken mainly around Beijing. But it was, so we had to also, based on the concerns of our Japanese community advisory group, we demanded new standards from Coriol that they should report who accessed the samples every 3 months. They had to send a report. Because initially Coriol repository said we don't want to submit, we don't need to give you a list of who applies for the research. What was interesting is the actual reason for this is probably partly the competitive researchers in RICN. They wanted to get the data of who was looking at their samples from competitive pharmaceutical companies. I wrote about this process in the because of some of the ethical concerns I had, although I was actually doing it, I thought it should be discussed. And so some of the words from the informed consent. So, donating today can be done anonymously. Your name or company name will not be recorded. This is what will be written. Blood tube, random 5-digit code number, gender, male, female, like this. The coding sheet kept by Eiko Suda in handwriting to be destroyed within 2 months. And the coding sheet was simply the number and the date it was taken from. No other information. No name, no location, and until this day we do not tell people where we took the samples in any conference presentation or anything. Please read the form, it explains the general protections. The forms will be kept by Tokyo University IRB privately. There's no individual discrimination. And why is there no individual discrimination? Well, partly we collected about 50 extra samples from to what we needed. So we randomly chose blood tubes to be made into the cell line. So if you gave your sample, you don't even know if your sample's in the collection in Coriol. Because we decided, even though it's a bit of a waste to throw away blood sample, this will be a protection of privacy. And the same was done in each country. So that people do not know, there's no stigmatization. You do not know if your sample is in the repository or not. Because you could look later, all the data is public. The genome sequence of these these samples will also be known. What diseases they have, and then somebody could say, well, I'm one of these people in the room who has this genetic disease, like the case I gave. But you don't know that, because you don't know your sample ever went to Coriol. In fact, in Japanese case, you do know if you were in the early round of sampling, your sample was certainly not there because it was contaminated. We had to do it in the second round. The budget was interesting. The budget for the ethics process in the end did not come from the grant. It was paid through somebody else's extra funds, which is sort of a little bit sad that it was still an issue here. The publicity, in the publicity it was said that if people from one area do not want to donate their blood, the project will not take their sample. The ideal community engagement process is where a community could reject donation. Since the Japanese government already gave over millions of dollars to the project, it'd never be an issue for Japan. In the sense that hubris about community engagement leading to possible rejection was false, at least in the Japanese case. I suspect it's also false in other countries who, despite the good sentiments of many, want governments will devote millions to a project not to have any samples. So this is sort of, I said that this is sort of a reality check for Ephesus, because in fact the government budgets are decided before the sample is going to take place, and so there's no way that a Japanese population was able to reject there being a Japanese sample there. Individuals could reject, but the government already had made this decision. This is another paper. You can find these papers on my website. Cultural differences in ethics codes are interesting. The Declaration on the Human Genome and Human Rights says no research could prevail over respect for fundamental human rights, fundamental freedoms, human dignity of individuals or, where applicable, groups of people. Now, the groups of people came in there thanks to a report I was a co-chair of for the UNESCO International Bioethics Committee on Ethics and Population Genetics, which was our response to a Human Genome Diversity Project proposals to do genetic sampling on tribes around the world. So you need to include human dignity of groups of people. How do you do this? Well, there are some mechanisms, but that's an interesting challenge. What views affect it? You know, if I were to ask, would you like to take your sample for predictive genetic testing? The accessibility of healthcare, prevalence of common genetic diseases, perceived susceptibility, income. These are all factors which may affect whether you want to give your sample and whether you would like to have predictive genetic testing. So for example, if I had a test which said, will you develop Alzheimer's disease next year or not? Would you like to know that? Who would like to know? Put your hand up. Who would not like to know?
Interesting.
So, this is a test which can be done. Potential benefits. Well, if your family had early onset Alzheimer's, which does exist, a few families have early onset Alzheimer's. You know, in the mid-30s, people get Alzheimer's. It's still in the reproductive age. It probably makes some sense for some people to know before they have kids. If when they're 30 years old, they're going to have children, and then 35 they're going to get Alzheimer's, they should make some arrangements for their children. Whereas if it's just general Alzheimer's when you're 65 or 70, then probably you don't care very much. Okay. Potential benefits of genetic testing. So you have choices. Relieve uncertainty of, you know, my parents have early onset Alzheimer's. What's going to happen to me next year? I want to know. This can help. Learning about beating a disease. You can learn, for example, particular genetic sequence. Okay. So sometimes, for example, if I have a cancer, do I want to get the genetic sequence of the tumor? And then I want to survey 1,000 papers in the medical literature to see which particular drug works best against this type of tumor. Okay. It seems reasonable. We may want to try and do that. On the other hand, some types of cancer are so complex that maybe just the general therapies may be all that we can go with.
And if someone was having IVF, they could have these tests done before they chose which one to implant. Right?
Yes, that's right. It's called Pre-Implantation Diagnosis. It's been standard since 1989. So for 26 years it's been clinically possible and offered in both in the US and the UK. So it's a whole generation of people with genetic disease have had this option in rich countries. And some have used it, and now some of them are considering for their children. What is a good phenotype? Is it bad luck or blame? Do I have guilt? In Japan, most people have a lot of guilt about having a genetic disease. They really think it's their fault, even though 1/3 of most genetic disease is caused by spontaneous mutations while you're an embryo. It's not even from your parents. but your parents will still usually feel guilty. That 1/3 of the cases of many diseases are spontaneous while you're an embryo. And then your mother might think, well, I drank coffee while I was pregnant. Oh dear me.
It gets really complex.
Yes. To change our children is changing ourselves. Genetic improvement. These are lots of questions. If I have a genetic disease, am I going to change it? General ethics questions. We're getting towards the end here. The extent of truth-telling is actually different. We talked about truth-telling in different classes. And if we're not telling people the truth, what about predictive genetics? We're not going to tell them the genes, what we're going to cause. But actually now our genetic information age is going to transform this, that people want to take control of their health. They actually will want to know, well, yes, in 5 years I'm predicted to get this breast cancer. So I'm going to take extra screening tests, or I'm going to
Remove my breast.
remove my breasts, even if it's a very low risk, and some people do. So how to handle this sort of information is very complex. And then we have a family decision versus the family risk and right not to know. I might have a right not to know, but if my child does a genetic test and finds he has a genetic disease of my grandmother, of her grandmother, my mother, I know I've got it. Unless it was passed through a carrier state. But for many genetic disease, if it's dominant, I will know for sure. So this is a rights to know. Example in Thailand of karma. Karma is common in many places. I'm not sure in Nepal if you believe in karma. So if somebody has a genetic disease, okay, it was my bad karma in my previous life. I was bad. It's my fault.
It's in most Asian countries.
Most Asian countries. So my concept of identity then, it's my own fault. And we have this, you think the Greeks have it?
I was just asking, do
Yeah.
Greeks believe in karma?
I think so, no?
Well, think about the Jews. The Jews, they have this biblical punishment that for generations and generations to come, your prodigy will be punished. That's sort of a karma. Yeah. So actually, it's sort of found in.
They probably believe in it.
Let's see. But you have good karma. Economic disparities. You know, all sorts of issues will affect the implementation of ethics guidelines. Many opportunities for exploitation as well. I'm going to capacity development is something. Public movements, anti-genetics movements are important. So genetics research is especially targeted. This is against genetically modified food in Japan, dressed up as GM corn. And financial conflicts and other issues. Well, anyway, we need to nurture the research by governments, academics, international research networks. So this example showed you some ethics statement by a human genome organization, which was a scientific organization. Now, some will say, why did they establish such a high-profile ethics committee? And they funded us to come to every meeting, all members. We could be very productive. Was it because they would be then seen to be an ethical organization? And to some degree, I would say yes. On the other hand, these leading scientists really wanted to be to show this. Last slide, I show you the human side of HCP. This is a slide I took in November. I was sitting here. This is Henry Yang. He's was the head of the Chinese Human Genome Project and still is. He's the head of the Beijing Genomics Institute. They have 6,000 scientists employed in 8 institutes in 6 countries around the world. They're all doing genomics analysis. 6,000. And their machines that they run now, they can sequence the genome in 30 minutes. Okay, that's why the costs are low. He runs this institute. Dr. Sakaki was the co-founder of the Japanese Genome Project. And in fact, we're making an archive, so all my old papers are part of this archive, since I was doing the ethics side of the Human Genome Project in Japan from 1991, sort of when it started, was doing the ethics side for 12 or 13 years. All these documents and the letters between researchers and the controversies, all these hidden secrets, we will describe because it's very interesting as an archive. This lady is the head of the archives at the Cold Spring Harbor Laboratory, Dr. Pollack. Jim Watson, her boss, could not be at the meeting. He prefers to be in the laboratory, and he's quite old. But he's still researching all the time, and we in fact watched his video, some of us, on why he did discover DNA and the genome. But making an archive then is very interesting. It's a public archive, and the Human Genome Project was sort of the second biggest international science project in terms of funding, and the largest in terms of the scientists involved. Does anybody know the largest scientific project in terms of funding? It happened before the Human Genome Project. It was triggered by a very famous president called JFK, who said, "We will send a man to the moon by the end of this decade." So the Apollo project was the largest international science research project in terms of funding. But the Human Genome Project is in the second largest. It was involved the largest number of people and the second largest amount of funding. These are tremendous science projects. Yeah, so it's a related current project, international project, which has not had, I think, so much success in generating global funding is the Human Brain Project, though I think intellectually it's a very interesting project. So big science, and therefore making an archive is very interesting. But when so many scientists, debated this project, it took so much money. So many scientists said, "Oh, I'm not going to have any money for other research." Everything went to the Genome Project. And it was the largest funder of bioethics research, still today, has been the Human Genome Project. So it stimulated bioethics very much. It stimulated how do we apply guidelines, and then how do we go back on some of these guidelines. Okay, so I will close for discussion and lunchtime or sleeping time, depending where you are.